Good morning, and the lecture of today's uh, the topic of today's lecture is pharmacodynamics. And as you know from the last week, the pharmacodynamics is uh, uh, is the way the drug will influence the human body. So today we will discuss how uh, how the drugs uh, make their effects, and the primary way the drugs will interact with the human body is interaction with the receptors. So most of the drugs are actually agonists or antagonists of the receptors, and there are a little one that will influence uh, the enzymes and will block or facilitate the enzymes and reactions that facilitated by these enzymes. And uh, this is the two most common types of the uh, actions of the drugs. And today we will mostly discuss the interaction with the receptors, because most of the drugs are usually act by by connecting with the receptors. So the receptor is usually located on the surface of the cell, and is a macromolecule that is usually a protein and has a large molecular weight. So this is a schematic uh, picture of the receptor. It so this receptor is has several subunits connected to each other. So it is a large uh, protein, and every receptor has several states. So and this is a schematic picture of the unoccupied receptor. That means that the drug is not connected to the receptor, and in these states the receptor will not uh, trigger anything or influence anything that is happening in the cell. So the receptor is empty, so that is why it's in a dormant state, like sleeping, and will not influence or facilitate any actions or the reactions. And so the next step in the activation of receptor is binding of the agonist of the receptor, and usually agonist is a drug. So this is a drug agonist that will bind to the active site of the receptor. This Y-shaped uh, part is a active site that will interact with the drug, and drug will be connected with this pocket, and that will change the state of the receptor to the active state. So in the active state, the receptor will trigger some intracellular biological response and reactions that will make the cell to do something, like, for example, to release the substance, or to start producing enzyme or to facilitate muscles to contract faster or any other biological response. So this is two states in which the receptor can be found, and then uh, there are different types of the receptors. So the main types of receptors are listed here, and the most common type of the receptor is line-gated ion channels, and ion channels are located in the membrane of the receptor, on the surface of the membrane, and they allow ions to pass through, like the sodium or chlorine, potassium ions. So every ion channel, they are usually specific, like the sodium ion channel will only allow the sodium ions to pass, and uh, this particular is a cholinergic nicotinic receptor that will interact with the acetylcholine and allow sodium ions to pass through the membrane. So when this receptor is open, the sodium ions will pass freely from the outside of the cell to inside the cell by the gradient of concentration, because there are more sodium channels outside the cell than inside. So, and this is actually acetylcholine that is connecting with the line-gated ion channel. The second type of the receptor is the G-protein coupled receptor, and adrenoreceptors are actually one of these most commonly studied receptors, and this receptor is more complex, has a binding site, and then has several subunits, alpha, beta, gamma, and the activation of this receptor will trigger the G-protein coupled uh, reactions that will inhibit or uh, activate the phosphorylation of the other proteins, and then eventually lead to activation of some reactions inside the cell. Then the third type is enzyme-like receptors, and insulin receptors are one of the most extensively studied ones, and enzyme-linked receptors will also lead to the phosphorylation, but they activated by the hormones like insulin, and uh, they will uh, they will lead to the change in the state of the enzyme that will facilitate the reaction. And the last type, the fourth type of the receptor, is an intracellular receptor. So all these three types of the receptors are located on the surface of the membrane, while the fourth type of the receptor is located inside the cell. It can be located in the uh, in the intracellular surfaces like the nucleus or uh, uh, on the surface of the organelles or floating in the cytoplasm. The protein, the result is usually gene expression or changes in the expression of the genes that will lead to the uh, synthesis of the proteins, new proteins, and some uh, whole body responses. So the steroid receptors or are intracellular, so they're not on the surface, and that is why the agonist should pass through the membrane, and usually steroids are very hypophilic, so they can easily pass through the membrane and activate the intracellular receptors. Sorry, and then lead to the activation of the transcription and translation, and the facilitate very slow changes in the body, but they usually very uh, important and will cover the whole body, because usually steroids will act through they will reach every tissue and part of the body through the blood. So the intracellular mechanism of the uh, activation of receptor is rather complex, but if we take as an example the G protein coupled receptors, so this is actually the receptor itself, and the agonist, or that can be a drug, hormone, or the natural neurotransmitter, then activates this receptor, and then this receptor is sitting near the G protein, and this is G protein that is receptor is bound with, and G protein will so this particular is stimulating type of the G protein. There are two types of G protein there. One is stimulating and another is inhibiting. So stimulating will activate the reaction of uh, synthesis of guanidine D-phosphase. So this reaction will facilitate, and so the G protein will start to produce GDP from GTP. So this process is rather fast and will take several minutes. And the next step will lead to the transformational changes of the receptor, and one of the subunits, alpha subunits, will disconnect from the receptor and move towards the another enzyme, adenylyl cyclase. So the adenylyl cyclase is a type of the enzyme that will promote the cyclization of the AMP. AMP is adenosine monophosphate, and cyclic adenosine monophosphate is a very important signal molecule, and the activation of this enzyme will lead to the production of the adenine or cyclic adenosine monophosphate, and that will interact with with the state of the cell and will lead to the relaxation of the muscles, so in most of the tissues, or any other response depending on the type of the tissue. So the once the agonist is disconnects with the receptor, everything goes to the previous state, and then the uh, the active uh, adenylyl cyclase is inactivated again. So, and then the receptor will rebind its alpha subunit back. So this is the way the cyclic adenosine monophosphate is produced in the cell. It's very common for many receptors like Agonists receptor and other other transmembrane receptors as well. So the enzyme receptors that is most common type is, so most intensively studied receptor is insulin receptor. Has also several subunits like alpha and beta subunits, and the insulin receptor is activated by the insulin. That uh, in after that it will activate tyrosine kinase. Kinases are also a type of the receptor, a type of the enzyme that will lead to the uh, phosphorylation of the tyrosine, and this receptor is actually bonded to the uh, beta subunit of the insulin receptor, and after the phosphorylation, this tyrosine kinase is an enzyme itself, and it will phosphorylate other proteins and then activate some signals. So, for example, it can inhibit the release of the insulin. So uh, the intracellular receptor is usually a large steroid receptor with a steroid structure. So this is a cyclopentan ergindrophenanchen drug. For example, it can be then like estradiol, and the drug or the hormone will pause uh, inside the cell. So this is a cell, and then will activate the receptor. The receptor usually is floating in the uh, uh, cytoplasm, and then it will enter, when activated, it will enter the nucleus and will react uh, with the specific genes. So this, and that will lead to the transcription and translation of the specific proteins, and then that protein will lead to some biological response. So the uh, the administration of the drug uh, in a sequence will lead to the reduction in the response eventually, because most of the receptors are limited and also have uh, the response once. So the second, when the drug is applied repeatedly, response will eventually decrease. So this is the feature for the most of the receptors, because they can decrease the number of the receptors, or the activity of enzyme will be limited because of the lack of the products, the substances that will react or will activate it in the reaction. So and a good example is the epinephrine. Epinephrine uh, uh, is used in emergency, and and also released by the body itself. So the once when you administer epinephrine first time, you get a maximum response. But if you administer just after that, after the response will decline, then you administer it second time, the response will be lower. So this so the so this is a very common for receptors. So because epinephrine will actually act on the alpha or beta receptors, and then to regain the response, then one need to wait for a long period of time. So so the receptors can recover from the previous activation. So for the drugs, to compare the drugs, the uh, pharmacologists will use dose concentration cues, and those concentration cues are actually depending of the effect from the concentration of the drug. So when the drug will start to act, the concentration is 0 and the effect is 0. So when we increase the concentration of the drug, then the effect will also increase, and this increase is exponential. So with the increase of the concentration of the drug, the response will increase to the some state, and at the um, higher concentrations, it will reach the plateau and will not increase further. So if we continue to increase the concentration but the effect will not increase, that means that all the drugs molecules are binded to all the receptors, so there are no more receptors left unoccupied, and that is why further increase in the concentration will not lead to the increase in the effect. So this is a linear response. So when the drug concentration is linear, and if we convert this graph to the logarithmic graph, so we take a logarithm of the drug concentration, this curve will become this type. The reason why we use the logarithmic, because it's easier to find out this 50% effect, because here it's hard to find the exact concentration of the drug, which will give you 50%, while here it's much easier to do that. So the EC50, or effective concentration, that produces 50% of the maximal response, is a constant for every drug. Like, it will not depend on the concentration on the drug or the dose of the drug. So actually it is the dose itself or concentration. So the lower is the EC50, the more potent is the drug. So the lower concentrations will make the drug more potent. It's not important it's not important clinically, but still the potency of the drug is calculated by the EC50 and is reverse connected, like the lower EC50 concentrations, lower EC50 values will give you a more potent drug. And so using the EC50, you can compare the potency of the drugs. If you have drug A and drug B, they're lower. So here drug A is more potent than drug B. Here also drug A is more potent than drug B. And the logarithmic curve is S-shaped. So this is S-like. So to find and usually this slope is almost straight, and that is why it's easy to find the 50%. So then the potency of the effect, so we discussed the potency of the drug, and then another important value, that is the efficacy of the drug. Efficacy of the drug is the maximum response the drug will produce, and it's more clinically important because the more efficacy the drug will give, the more is valuable to the patient. And in these examples, drugs A and B have the same efficacy, while drug C has less efficacy. And also, if we compare EC50 is lower for drug A, so drug A is also most potent drug, and then B is less potent and C is the least potent drug. So these are differences between the linear graphs and logarithmic S-shaped graph, and will show the amount of the receptors that is bound to the drug, to the divided by the total amount of the receptors. The if we use with the if we look on the uh, the dose dose or concentration response uh, uh, graphs, then we can, by looking on the graph, we can identify the type of the response from the agonist, antagonist, and also the types of the agonist. So there are three types of the agonist known, and the first is the full agonist. Full agonist is most effective one and will lead to the maximum 100% response. So this is usually the most common type of drugs. Then the partial agonist will lead to the less than 100% response even in the maximum concentration. So the partial agonists usually have some antagonistic action and lead to the activation of the receptor with the incomplete activation of the receptor. And then there are also inverse agonists. Inverse agonists will lead to the decrease in the response of the receptor. And if you combine full or partial agonist with the inverse agonist, then it will reduce the efficacy of the full or partial agonist. Then when the drugs are administered together with the antagonist, so there will be two types of the antagonist. So this red is drug alone, so and it is a full agonist. So when you combine a full agonist with a non-competitive antagonist, it will make it a partial agonist, so the response will not be full, but the Q will stay here. When you combine a drug with a competitive antagonist, then the Q will shift to the right. So this Q S shaped and this is also S shaped, so they are identical. But now with the competitive antagonist, the drug will require higher concentrations to make a biological effect, and that is why the concentration response curve, this curve will shift to the right. So this is called a right shift and will increase the EC50, so the drug will be less potent when it's taken with the antagonist, because competitive antagonists will bind to the receptor, but when the dose of the agonist will increase, the agonist will take the receptor back. So competitive antagonists will compete with the agonist to bind to the receptor, so the higher concentration wins, and then that is why this Q is same. So the agonist will still have the maximum effect, but the higher dose will be required. And the last the last parameter that is important is the therapeutic index. Therapeutic index is the range of the concentration where the drug is safe, and so the low therapeutic index will mean that the even slight increase in the concentration will lead to the toxic or undesired effect. So this cube is the therapeutic uh, therapeutic uh, effect, and this Q is adverse effect. So you usually don't want adverse effect to happen, or toxic effect. So, and this is the concentration. So, and in this, so this is warfarin. Warfarin is the anticoagulant that is used after the surgery to prevent the blood clot. So, but the undesired uh, effect of the warfarin is the bleeding. So actually this is a blood clot reduction, so anticoagulant effect, and this is a bleeding. This is red. This is what this is red, because the patient will bleed and then will lose the blood and die. So that is why physicians usually don't want this to happen. So that is why, but when they sometimes when they want a higher effect, they increase the dose, and in case of the warfarin, the maximum dose of the desired effect is uh, overlapped with the uh, undesired, the a little bit undesired effect. So that is why patient will suffer a little bit bleeding here if you will increase the dose that high. So if you stay here, you are fine. So this is a EC50 actually. But if something happens and the dose will increase slightly, then you get a bleeding. So, and then you patient goes to the emergency room and surgery, and so emergency room and the ambulance. So that's, you don't want this one. So this is a low or small therapeutic index. Therapeutic index is actually the uh, is the concentration of, so the dose or concentration, the dose of these divided for dose of these. So if it's low number, then even slightly changing the dose will lead to the adverse effect. So another example is penicillin. Penicillin has a large therapeutic index, and it's seen here that when we increase the dose and we get the maximum response, we still have a window, so to and even we increase a little further, we still will not get the unwanted adverse effects. So uh, that that so that means that if we divide this dose to this dose, we get something like 10. So we actually it's 1000. So you need to increase the dose 1000 times to get some unwanted effects. Actually, I think even 10, yes, 10 5 times to get them even small unwanted side adverse effects. So, but this drug has a large difference between the doses for the desired effect and unwanted adverse effects, and that is why the therapeutic window is a large or wide. So this is a low, and this is a wide therapeutic. So the large, small therapeutic window and large therapeutic window. So window is actually the range of the doses that can be used to obtain the desired effect. So thank you for attending the lecture, and what are your questions with uh, for the pharmacodynamics? No questions?
I think so.
Then let's make a short break, or